currently pursuing Masters in Texas University, Kingston. I am working in Dr. Perez Balston's lab for my research projects. Our project is bioinformatic tools for influenza A virus, genome sequencing and comparison. The main aim of our project is to use various bioinformatic tools which are available for genome sequencing of influenza A virus and comparing those genes with other subtypes using some bioinformatic tools like BLAST. To know our project, you guys need to know some basic terms like what is bioinformatics, what are bioinformatic tools, what is influenza virus, how it causes, something like that. So I will briefly explain what is those things. Like first is, what is bioinformatics? So bioinformatics is the application of computer technology to the management of biological information. It was like, these guys, like it's a mixture of statistics, biology, and computer science. So if you mix them, you will get the bioinformatics. Something like, it's a mixture of subjects. And it is essential to the use of genomic information in understanding human diseases, like and also in identification of new molecular targets for a drug discovery. So it has vast applications. And bioinformatic tools. The bioinformatic tools are software programs which are used for saving, retrieving, and analysis of biological data and extracting information from them. Some general used tools are like BLAST, FASTA, EMBOSS, CLUSTAL-W, Plasma. Like a BLAST means those are all of the programs to analyze the sequences. I will tell you briefly about BLAST. Like we used only BLAST in this project, and this BLAST is like a basic local alignment search tool. It is used to compare local genome sequences. And FASTA is a text-based program like representing nucleotides and peptide sequences. EMBOSS is European Molecular Biology open software suite, and CLUSTAL-W is a program for protein multiple sequence alignment, like different multiple sequences to analyze those. CLUSTAL-W is used there, and RASMOL is a program for molecular graphic visualization. Next, guys, we deal about influenza virus, right? So influenza virus influenza is an infectious disease which was caused by influenza virus. Influenza virus is a member of orthomyxoviridae family. There are basically three types of influenza viruses: A, B, and C. Like A affects humans, wild or domestic animals, wild birds. Like the B is in nursing homes, like sporadic. Some deals with that. It affects nursing homes, like and in influenza C affects humans and pigs. It was lesser extent compared to A and B. And like the flus, these viruses are responsible for various flus like avian flu, horse flu, swine flu, etc. And we are restricted to only avian flu in this. In this avian flu is avian means bird. So avian influenza is an infection which is caused by avian influenza virus. There are many subtypes in the avian flu, like H7N7, H5N1, H7N2, H7N2, all this. And the main focus of our project is on the bird-adapted strains of H5N1 influenza virus. So we are restricted to only H5N1 in our project. So this is a basic structure of influenza virus, guys. You can see these are RNA. This is negatively coiled single-stranded RNA, which has 8 segments. In these 8 segments, these 2 segments, hemagglutinin and neuraminidase, are main focus of our project because those are responsible for its virulence nature, hemagglutinin and neuraminidase. I will tell you about all the 8 segments in the coming slides. And the causes, like avian influenza virus infects pigs and mixed with pig influenza virus. Then the virus exchanges genetic information and forms a new virus. This new virus affects the humans and easily spread from person to person. So this is how it infects. And the symptoms always depends on the strain of virus which is going to affect a human. Like normal symptoms are cough, sore throat, fever, diarrhea, all this, malaise, headache, etc. And the disease mechanism. So basically there are main 3 steps. First step is how the virus is enter into the cell, and second step is where the genome is, like where the genome is where it delivers its genome such that it replicates its new viral copies of RNA and proteins. And finally, it should exit the host cell to infect other cells. So I will tell all the 3 steps in this infection cycle. First is this virus should enter into the host cell. So this is influenza virus, it should enter into the host cell. This is our first step. How it enters means the hemagglutinin present on the surface of the hemagglutinin here is present on the surface of the virus is going to bind with sialic acid present on the surface of the host cell. And the neuraminidase, it binds to glycoprotein and serving to free hemagglutinin binding sites for the surface receptor. So binding tends to form an endocytosis, and which tends to virus enters into the cytoplasm of the cell. Now there are also M2. I told you guys, like, there are 8 segments present on the surface of the virus. One of the segment is M2 proteins. So these M2 proteins allows the protons present in the cytoplasm of the cell enter into the virus virion. So we know the protons. Protons makes H plus ions. So those due to the increase of H plus ions, the acidic nature environment increases, and this leads to formation of peptide, fusion peptide. This fusion peptides allows the viral RNA present inside the cell to deliver outside, like this viral RNA escape from the virus into the cytoplasm of the cell. So when the replication starts means when this RNA gonna access to gain the nucleus of the host cell. When it gains access to host cell, then the replication starts and multiplies and forms new viruses and extends to other infects with the help of budding like this. So it infects other cells. Next is methodology. This is all the basic things which you have to understand. Now the game starts. So this is strain H5N1. We extracted, like this is a strain name A/TK/Inan. Actually it was extracted in China in 2004. So we took that virus. Like I already told you, it's a member of Orthomyxoviridae family. It's negatively coiled, single stranded. It has 8 segments. These are all 8 segments. Hemagglutinin, matrix protein 2. We see these 2 proteins and neuraminidase. And polymerase basic protein 2, polymerase basic protein 1, polymerase resting protein, and the nucleoprotein. These are all 8 segments of proteins which are present on the surface of the cell. And what we did means we want genome sequences, right? So to get the genome sequences, we need action numbers. So for these action numbers, the action numbers of the genes for all the 8 proteins, which shown in the last slide, were obtained through biohealth base. So from the biohealth base, we got the action numbers. So using those action numbers, we got nucleotide sequences for all the 8 segment proteins. So from where means it is from NCBI. NCBI is a National Center for Biotechnology Information, where the nucleotide sequences are searched for a given action number. So it is readily available. If you have action number, it will give the nucleotide sequence. The sequence comparison, actually we can compare any kind of we can compare chicken, the same virus which affects chicken, and same virus which affects swine, or like we can compare 2 different birds, or we can compare 2 different subtypes. Like in the same chicken, we can apply H5N1 virus, and in another chicken we can apply H1N2. Like we can compare 2 different subtypes. You can indicate like influenza A virus on one thing, influenza B virus on one thing. So we can compare whichever we can. So in this project we did 3 comparisons. First is we took H5N1 influenza virus, we infect chicken and swine, same virus, and we observed neuraminidase gene in that. And the second comparison one is done in polymerase basic protein 2 genes of H5N1 and H7N2. So So here we are observing two different subtypes on same bird, chicken. Here we have two different birds, chicken and swan, but we are applying same virus. And in third comparison, we took hemoglobin gene for influenza A virus and B virus. So we are taking two viruses here, two different viruses now, A virus and B virus. There are lots of drugs for influenza viruses like amantadine demanding for A and Reliance and Tamiflu for A and B. And Tamiflu is like it prevents this. Tamiflu acts on a neuraminidase enzyme and it prevents the spread of infected cells to other healthy respiratory cells, and thereby it decreases the infection process. So its limits. So finally we got results like our project is like 40 pages. In that the 20 pages are results. Like all the 8 like hemoglobin sequence I showed. This is an example like this. So we got lots of sequences for hemoglobin, lots of sequences for all the 8 proteins. All the proteins we got lots of sequences, and these six sequences were with the help of action numbers. We got sequences from NCBI. Already mentioned here about that. And what we got the result is in the first comparison. So I have compared chicken and swan infected with H5N1. I got 95% similarities. So 95% identities were found between them. And the second one, I got 83%. This is only chicken, but I applied two subtypes, H5N1 and H7N2. Two different subtypes. On chicken, I got 83% similarity. Even though in the third comparison, even though it was hemagglutinins, which is mainly responsible for virulence, but I got no similarity. We got no similarity. No significant similarity is found because they are completely two different viruses, A and B. And finally, I can conclude that there are minor variations within a particular class of genes only. And the variations in a particular class is only in few genes. It was quite confused, like simple thing is like for example, when hemoglobin genes of H5N1 and H7N2 are compared, like in the second comparison, we got, how can we go back?
Oh, with the arrow keys, the arrows on the keyboard.
Keyboard, yeah. Like in the second comparison, guys, I compared two different subtypes, but I got 83% of similarity. So there was only little difference in both the subtypes, like only few of deletions in the sequences which leads to another subtype. So like that. And finally, like as a majority of genes are in all the subtypes are similar, like we get the maximum similarity, like 83%, 85%. We get the broad range of drugs are in use for all the influenza viruses. There are lots of drugs we can use. I would like to thank my mentor, A.C.P. Chaudhary, sir, for encouraging me to work in Bharat Buddies project. And I would like to thank Dr. Perez Balisteru because for allowing me to work in your lab, irrespective of the background I have. And thanks for giving me Sarah, such a good mentor. I love you, Sarah. And these are the references. Like there is no chesting, no questions. Hopefully. Thanks.
All right.